The broadcast is now starting. All attendees are in listen-only mode.
Good morning. Welcome to the ILSOI Advisor webinar on soil quality, soil health challenges and measurement, brought to you by the Illinois Soybean Checkoff. My name is Kendall Brisketall, and I will be moderating the webinar today. A couple of housekeeping items. If you included your CCA number when you registered for this webinar and you stay with us for the entire presentation, your number will be automatically submitted for 1 CEU in soil and water management. If you are listening to a recording of this webinar, you will need to go to the Certified Crop Advisor website, log into your account, and apply for a self-study credit. There will be an opportunity to ask questions during the webinar by submitting your question using the chat feature. There will be a time for question and answer at the end of the presentation. Please keep questions brief and only to one point. Without further delay, I'd like to introduce Will Brinton with Woods End Laboratories of Mount Vernon, Maine. The presentation will start with Will. Take it away.
Thank you very much. It's a very wonderful time to be giving a webinar, and I'm grateful for the Illinois Soybean Association for inviting me in. Today we have a challenging topic, an interesting topic, and a controversial topic to present, and I'm really looking forward to sharing my thoughts with you and the work that we've been doing. Our business is Woods End Laboratories. This is a company I founded for soil testing when I was an undergrad in agronomy school. So I've been at it quite a while, and I've watched the field develop and change over a period of time. And in the 90s, we had soil quality and soil quality indexing, and it somehow morphed into soil health and soil health testing after the 2000s hit. So this continues to be an open discussion, and today I will focus in on some of the challenges that we are experiencing and some of the excitement as we go forward. I want to begin by making a statement that I think is obvious, but sometimes we lose sight of. Soil cannot be healthy by itself. If you think about when we talk about soil health, it sounds like it's a thing in itself, but left to itself, soil may become eroded or even desert. And we know this is one of the great crises of the world, is the increasing amount of deserts forming from unmanaged and uncared for soils. But when I say it can't be healthy by itself, what I mean is that it is the plants and microbes and animals that participate to create and sustain soil health. As a picture here on the right dramatically shows, there's a rich layer of soil on the top. The reason it stays fertile, the reason it stays high in organic matter is only because of the management practices upon the soil surface, the presence of plants, the presence of soil animals, and larger farm animals that maintain it. And on a really positive side, and I make this third point here, farming has shown itself capable of preserving and augmenting soil fertility over hundreds of years. Now, the picture on the right, I found on a Belgian website of farm soil that has been continuously farmed for 800 years. And it's really impressive that if we do the right things, we can maintain, augment, and preserve fertility indefinitely. Having said that, let's look at some of the details that are involved here. I like to point out that health is a result of interactions of many different functions forming a complex whole. So any way you want to look at it, there's a number of different details and angles that become important. And I think one of the most important things is to start looking at the interactions. As the image on the right shows so dramatically, you almost have to look at it and say, what am I looking at? This is a soil in south central Pennsylvania. We dug this pit a couple years ago, and we were astonished to see the evidence of soil building going on. Now, these dark streaks in the soil here, these are earthworm canals from previous years, and this is probably just from the recent year. You can see an earthworm curled up over in here, deep down. This is about at the 2-foot mark in the soil. And notice what's happening. The roots are following the channels down. And if you like to read papers on earthworm castings, we all know that they are substantially enriched in nutrients and carbon. So it provides the roots easy fertility, and it provides water access to the soil, and it's decompressing soil by opening up pore space. So right there is an interaction effect that's difficult to quantify. In fact, I like to point out that we don't know what are causes and effects here. I've shown this picture to audiences, and some people feel that the root was there first, and others say no, the worm was there first, and so on. So this is a fun topic. It involves every aspect of biology, chemistry, and physics that you want to put in, and it even implicates the entire climate, as you can see indicated here. The whole exchange, the gaseous exchange from soil to air, is one of the primary features of characterization of this system. So let's stand back and look at analysis tools. And my point about this complex system is you have to select a tool in accordance to differing functions, and these tools are very different. People ask me, what is a test for soil health? I say, well, which area are you looking at? There are so many different angles to this that you can just focus on looking at food, carbon as foodstuff. That relates to primary and secondary decomposers in soil. And we hear so much about bacteria and fungi and AMF, but people forget that a host of soil animals are doing the primary decomposition and are very, very significant. And methods to characterize and measure them are difficult and very, very rewarding. Then there's a whole subject of nutrient and organic matter transport, and what a stunning image of organic matter transport we have right here. So one of the hallmarks of soil building is that fertility is being transferred from the topsoil into the B horizon and further down by the action of plants and animals. And this leads to structural and infiltration issues. This is a whole field in itself. There are many different ways to measure it. And finally, coming back around the circle, the respiratory carbon dioxide and oxygen influx and efflux is one of the most significant features of the whole system. It is not static. There's constant air movement in and out of the soil. And I have made it a focus in my life to be able to characterize particularly the carbon dioxide relationships and exchanges. Let's touch on some of the issues that I will be going into today and get that out there. I like to look at how soil testing affects the practice of farming and how soil health affects the practice of science. And both of these areas are challenged because science is part of the problem too of soil degradation as well as farming practices. Each side of the equation must be dealt with. In the area of farming, we have questions such as, are analyses relevant at the magnitude of farming practices? So you can do minute analytical procedures, but it's difficult to know how relevant they are. And closely related to that is, can results of soil health testing be interpreted into management decisions? I often get asked about some of the procedures that people haven't seen before, such as PLFA. This method has been around for more than 25 years, and it provides a very interesting description of the potential presence of different organisms in the soil. But in 25 years, we have not seen a single means of interpreting that method into management decisions. And I will explain this later. So analytical methods can be excellent, but be challenging to turn them into practice. Now let's look at the practice of science. One of the areas I've been focused on in the last 5 years is this enormous significance of when you're measuring biology or ecological systems. Your measurement of it and the way you handle it is a disturbance of the trait that you desire to measure. It's called disturbance or artifact production. And we have found that every aspect of sampling, drying, and grinding soils is implicated in disturbing the system we are trying to measure. I have recently done a literature survey going back 45 years of studies related to the disturbance in the laboratory of the biology being measured, and we will be putting out an interesting technical memo in the near future about how important this is. We have developed soil testing for nutrients. We have developed soil testing for physics, but the practice of handling soil for reproducible biology testing is more challenging. The other big concern in science is, are we converting results into fertilizer rates? Is that our only interest? Or are we trying to convert results and build soil? In other words, find ways that the indicators can simply help growers understand we're doing things better. If we're converting it into fertilizer rates, it's going to put a lot of pressure on us for accuracy and precision. But if we're converting it into soil building practices, that's a different matter. We just need broad brushstroke indicators that work well and correlate. And here's a big area. We need to adjust our indicators for soil physiographic regions. This is one of the most important things. We simply can't approach this subject, I believe, by saying we have one soil test for the country. We have to look at it in this sense. Here is the geophysical provinces of the United States, regions we know well, and all our soils have formed differently in these different zones. It's very, very difficult to imagine scientifically that we could create a single test that works in all these zones, particularly when you look at the soil orders. So the soil order maps for the country closely follow these ecophysiographic zones very naturally, and we see the whole center part of the country with these wonderful mollisols blending into the alpha soils, and then the southeastern soils, the Utasols and Piedmont soils. Every soil has characteristics that are very, very significant, and each kind of soil order will express itself through soil health metrics differently. If now you take and put the climate on top of it, look, notice how in America the climate is really a horizontal feature of the landscape, whereas if you look back at the soil orders, these are almost vertically formed in relationship to the layout of the land physically. So the climate intersects all these different soil zones and changes the quality of them significantly. So the work that we have been doing in the last year is building this model into our soil test results so that we can pinpoint where you are both in your climate zone, rainfall zone, and soil order and suborder to begin to make sense of interpretation of soil tests. It's proving tremendously challenging and extremely interesting. So coming back and summarizing this introduction, healthy soil arises out of the intersection of soil formation forces, soil parent material, climate, seasonal factors of temperature and humidity. And really, it's what people decide to do with these parameters that makes all the difference. This is the model that we use here at Woods End Laboratory to describe this interaction of physical, chemical, and biological forces. Where they converge, we like to say that's your soil health, but it's not necessarily true that the convergence is equally balanced between all these factors. It depends what kind of situation you have. So another factor here is farmer innovation with soil health cropping. I like to call it soil health cropping, is significantly increasing the complexity of analysis. So now we're dealing with multi-crops, multi-seasons. So what's the right sampling time? This is a question I get asked frequently. And often the ground was bare in the fall after a crop. You could go out and get a clean soil sample, but no, not anymore. It may be well on its way with a cover crop coming up. An amazing thing that we have found in analysis, increasingly as we diversify farming with more cropping, we're seeing a lot of biomass nutrients that are not going to be revealed in the soil test. So at any given moment when you test your soil, a whole bunch of nutrients are not in the soil, but will be again when the plant decays. The other thing that this relates to is this significant feature that carbon biology is a delayed effect compared to nutrients. You can put on nutrients, test the soil 3 days later and detect them, but carbon biology is not like that. It's the delayed effect part of it that's challenging in measurement, because we might put litter into the soil or on the soil and not see the respiratory effect of that for weeks or months. And this is one of the areas that needs quite a bit more work. So here's all the indicators. all the different zones that you might want to zero in on. And we have, in order to build our soil test, we've gone around and picked out items that we feel we can reliably measure, and we've developed testing around them. And it's important when you do this to try to encompass the entire circle. We all have different disciplines and training, and we might want to zero in on something that we're particularly good at. And I have tried a lot of these things in the past, and gone from one to the next, and arrived at a way that we can make it work a little bit better. But again, I believe in multiple indicators, and there's many different ways to skin the beast here. So I want to make another very strong point. Long-term field studies are absolutely crucial for interpretation and calibration of soil health testing. I got my whole start in my career by being invited to be a grad student in Sweden, assessing a 17-year-long field crop study that was coming to its end. Actually, the study got continued, and somebody came over to Tufts University several years ago and gave the 42nd year report on the changes in soils in this long-term study. I went down recently and visited the old rotation down in Auburn, and it's an amazing fact that these long-term plots have been there since 1896. Here's an example of cotton grown without any fertilizer, because every year they had winter legumes in the rotation. The fertilizer was only put on the winter legumes, and the crop did well after it. Neighboring plots on the same soil have very, very low fertility that did not have the cover crops. So what we are doing is we're accessing some of these studies. Rothamsted in England is well known. Auburn, I've mentioned, the Morrow plots in Illinois, the Sandburg plots in Missouri, and these ones that I experienced in Sweden. And I know the folks down at Auburn University have used these plots now to help calibrate their own soil health tests, and that's a wonderful way to do it in a very meaningful way. Here's another really amazing example of how much we actually know about soil health indicators. This was a study published last November, a worldwide survey of paired scientific studies comparing soil health indicators, where you had paired comparisons of organic versus conventional management. The authors were very discriminating in which studies they picked. They discarded any studies where the pairs were unequal in some way, or the soil types were different between the pair, or the management was so different that you couldn't really compare the two farms. But look at what we've learned with multiple indicators. This study shows that 41% of farms had more biomass carbon, 51% more microbial nitrogen, 59% higher PLFA counts, 74% more dehydrogenase. Dehydrogenase is an important enzyme that uncouples organic matter and helps the decay process begin. 4% more efficient CO2 cycle. That means less respiration per greater amount of nutrient release. 84% greater protease, 32% more urease. These are exciting features. But in other words, what I'm saying is we already know a great deal about indicators when we're doing comparative farm analysis. This is an example of the early work that impelled me into the field of respiration and soil quality. This was the 17-year study that we did measurements on comparing soil respiration and earthworms in soil. And these were all the different treated plots that had been managed by different rotational systems over 17 years. And we had an astonishing correlation of respiration with earthworm canals. Now, that doesn't mean the earthworms were making the CO2. It means the association of worms in soil bacteria were causing this. It was amazing also to see the control soils had stayed at a certain location, whether we did the one-day test or the one-week test of respiration, we got the same results. And we were using these good old-fashioned methods here, titrometric base trap CO2 respiration, a method that was standardized in Germany in the early 1950s, a clumsy, time-consuming procedure. And that was where I resolved to develop and evolve a new method. And as we developed this new approach of gel technology to trap CO2, we went out to other groups, and And I want to acknowledge the work of Purdue and Iowa State in helping me in some of the early studies in validating the measurements and how they were being performed. We had astonishingly good results in the optical system that we use. We use this color chart, which this is almost an exponential scale, like the pH scale, in correlating that with the amount of CO2 per kilogram of soil. But even better, people ask me how precise and accurate is the chemistry of our test. This is a calibration assay done with our color system compared to calibrated gases that Iowa State did. So very, very high degree of correlation. So we know the potential is there for accuracy, but the tricky part is how we handle the soils. The next thing I wanted to do to build more soil tests, measuring features that we're not commonly measuring, is to find out how we can get to amino nitrogen in the soil. Amino is this enormous store within organic matter of nitrogen that's potentially available. And I developed a chemistry system for that. You put soil in here, you release the amino compounds by adding a reagent to it, and it colors the gels differently. And it's an exciting visual way of looking at it, but it's really important. For example, here's a virgin prairie soil from Nebraska that had 200 parts per million of color in it. And here's a depleted soil in North Carolina. And nearby neighboring soil, Ray Steyer's no-till cover crop farm, he has taken a soil that has been depleted completely of organic nitrogen and put all that back into it over 30 years. The validation studies on this have been ongoing over the years. Particularly want to acknowledge the work at University of Connecticut that we have collaborated on. We're getting really excellent correlations using the amino N test and yield of turf, for example, in this. This is working particularly well when we're using organic fertilizers. This study was done with a product out of Minnesota. It's really important to understand with soil health measurements that we're trying to build an idea of the probability that with good soil health, you won't need additional fertilizers. And I really am pleased with this probabilistic approach that the University of Connecticut has applied, and this is in David Moore's recent master's thesis on it. So this is our soil health indicator test across the bottom here, and this is the probability of that indicator exceeding a benchmark set by different rates of fertilizer. So if you only want 50 units of nitrogen, at this level here of 140, you would have a nearly 90% probability that you do not need any fertilizer if that's your soil health. But if you need 100 or 200, you can see how the curves shift. So soil health is one thing, but correlating it with what we actually need for our crops is a quite different matter. So you might be really disappointed in this value if you're way out here needing 220 units of nitrogen. This is the black curve here, and you're just not there. You need to get your probability up, and we have found when you're in 2/3 level up here, you have a really good opportunity to start pulling back on your fertilizer. I've done quite a bit of work in Illinois, thanks to Richard Mulvaney's group in Urbana, and I wanted to show some really great correlations we're getting comparing methods. Now, we developed this new SLAN method, and there's the ISNT, the Illinois Standard Nitrogen Test, that was developed by Dr. Mulvaney and has been used quite successfully in a number of regions of the country, even outside Illinois. But what we did in this study, we took different soil series from the state, and we've been working with these different soil series here, and found this very, very high degree of correlation between our new amino test and the ISNT, which is more of a standard. But look how strikingly different some of the soil series are. I put these green stars here. These are two soils that have been found to be non-responsive to additional nitrogen. This one particularly, you can almost see why. Look at that tremendous range of organic nitrogen compounds. Here's a soil that is responsive, the Cisna series, and it has a much lower level of these factors present and much more scatter and spread. So we know there's some very interesting things there. So then I said, well, how about carbon? So the exciting thing here is we took the same 4 soils and correlated our tests with total soil carbon across a range all the way. The highest level were on the Flanagan soils. We had up to 3.5% carbon. So that's a 7% organic matter soil. Really astonishingly good correlations. So what we know is that the carbon is the repository of these amino compounds in soil. Now, one of the things we had actually done here, and the reason you see groupings of soils here, is we did 3 different profiles and compared them. And here's what the state looked like in these 9 different soil series, with topsoil, mid soils, and subsoil in each category. So what we call here deep, mid, and top are these 3 soil samples. And it looks like the non-responsive zone is somewhere around an indicator of 150. So here's your Flanagan soils. Both the topsoil and B horizon are rich in amino compounds. No wonder these soils are less responsive to nitrogen. But here's the Cisna soil way down here. So we're able to use this method of soil health testing to start gaining a sense of the behavior of our different soils. And if you add up the amino compounds from 0 to 6, 6 to 12, and 12 to 24 pounds per acre, Flanagan soil, 1,100 pounds an acre of amino compounds, whereas Cisna had only 300. Using the organic nitrogen index system that I use, based on what the mineralization of organic nitrogen compounds is, we would likely see this kind of difference in nitrogen released kilograms per hectare under these different systems. So astonishing different behavior. So here's the whole group of soils correlated now to microbial respiration across the state. A very, very respectable correlation, but ranging though from very low to very high across all soils and depths. So we know there's a tremendous variability in soil health indicators from low to high. So what I did here is break it out now into the topsoil, mid soil 6 to 12, and 12 to 24, and you see how distinctly these populations break apart. So now we know all the reserves are here in the topsoil, but the slope here, the correlation of these is much lower, which means it's going to be more difficult to use the topsoil alone to relate these 2 indicators. But if you add the subsoil into it, you get a much more satisfying response. All these were very highly statistically significant considering the quantity of samples. And we're wondering if a model might be to use the amino N and microbial CO2 rates at both depths to provide even more substantial evidence that of responsiveness or non-responsiveness. And clearly we think the rooting depth and moisture access is going to play into this. So here's another study we did in Canada, finished it last year on 15 farms, and what we were comparing in collaboration with A&L Canada's Bioresearch Division is, can we use NDVI imaging on crop stress and relate it to soil health indicators? And the summary, this is just an example of an NDVI image where you see a stressed area in the field. This happened to be a wet area and a very healthy area. We would go in and take 10 samples from each of these zones and then run the analyses. And when we compared overall all 15 farms, green versus red zone, stress red, non-stressed, our soil health traits were significantly different between them. So it's not just nutrients that are causing these stress regions. There's something going on with soil health in those zones as well. A test that I've been working on developing, obviously to complement these biochemical tests, is we have to get at the soil physical structure. And many of us have seen these popular demonstrations of putting the clod in the water and watching it decompose. It's usually a pretty stunning test when you do it. One soil will sit there and not fall apart and the other will. It is somewhat of a misleading test because if you allow the soil to get too dry, and I've seen many demonstrations where they use a dry clod. What happens is the force of water diffusing into the soil and causing swelling, sudden swelling of dry clods, will cause them to explode and fall apart in this test. So it exaggerates the differences between soils when you do it with dry soil. We developed a sieving method, which we call VAST, volumetric aggregate stability, that's very visual, rapid, and is giving very reproducible results, where you start with a soil and you start dipping it, and you get to a point where on the screen is all the stable soil that's forming aggregates that will not disperse. And then you disperse the aggregates physically, and what's left is the substructure of the soil that appeared to be aggregates, but in fact is only sand particles and grit. So our test uses the geometric differences between these areas to produce a meaningful result. I want to talk a little bit now about all these different soil health scores, and we see different labs are in the field. I've done a lot of work with this. This was a study that Dan Davidson helped us with, and he reported on last year in North Dakota, these findings. So what we did was Dan had selected 6 different farms of known different characteristics. He knew what to expect, and he sent it out to our lab, to Cornell, to Ward, and Midwest, and we all ranked it with soil health tests, and I put them all up on the chart here. Everybody agreed these soils were in the depleted category of low soil health, and we came very close to agreeing on all the very best soils here. And then there was these middle zone soils that were somewhere in between. But if you do rank order correlation, and that's the right way to do these kinds of studies, everybody tended to agree on the best and worst farms, with a little bit of overlap on the ones that were the kind of management that you're not sure if it's could be better, but it's doing well. And I thought, well, this is really a good sign that different approaches to testing using rank order correlation show that we are doing a pretty good job distinguishing soils. So we then took those soils that Dan had prepared, and the most astonishing correlation, usually you don't ever get correlations this good, was the Woods End comprehensive test correlated with the Cornell test. And what we did was use the overall rating scores from both our systems. So these are 2 labs that have taken different approaches to soil health in the last 10 years. And we have overlapping tests, but they're differently organizing. But when we ranked all those 6 farms, we got nearly perfect correlation. So the good news is a lot of these tests can have differences, but through rank order correlation, we can conclusively say we are able to distinguish a range of fertilities and soil health factors with these procedures. And I like to emphasize this because so many critics are saying you have to be precise and accurate, but this is how you get to it, through rank order correlation. It's not the exact number, it's the capability of distinguishing differently treated management systems from each other. Here's a study that was really important to me and now is ongoing. In the Northeast here, we decided we have to ask the question, what is soil spatial variability doing to soil health? It's the dreaded topic of all soil testing is how we sample soils. So the question we asked, how does sampling affect nutrient tests versus doing some soil health tests? And they chose to do the CO2 burst and the SLAN amino N. And then the question was, what is the variability of lab analysis of common nutrients versus soil biology tests? The hypothesis of this study was that soil biology tests were fundamentally variable and much more so than common nutrient tests. The cooperators are listed here, and we spread out all around the Northeast on 17 farms with 4 different means of sampling each of the farms. So here's an example in the picture here. This was one mechanism sampling called the individual core approach. That 12 cores taken in one location in the field, that as one possible way of representing the soil. And then we did random, where you walked all across the field and took 3 samples of 12 cores each, plot studies where you select a 40-foot grid, and then between row versus in row testing. So all these results were then, soils were coded and sent to 2 labs. Woodson Laboratory did all the biology, and University of Connecticut did all the nutrients. So here was the astonishing result using the random model of sampling to do our statistics. These are all the variables that we tested. And these 2 columns here give what the results were. To attain 20% precision in testing, and we decided 20% precision is, we believe, is the best you can really get with soil testing. You needed 1 sample for pH. That is one sample consisting of 12 cores each, which was 9% precision. And the numbers climb progressively here. So phosphorus, for example, was so inherently variable that you would need 33 samples to get 20% precision with an overall 94% variability of 1 sample. Again, 1 sample was 12 cores. So astonishingly, the respiration and the SLAN were the second lowest variability of all the tests, certainly compared to the nutrient tests. And even the VAST test had reasonably low sample requirements to attain satisfactory precision. But if we're only doing 12 cores each, this is what we have to expect. 20 to 30% may be the norm, and I think it's realistic to approach it like this. Now, one thing I will say, pH is often used as a comparison for variability, but it's really unfair comparison because it's already a log transformed value, which means it's a compressed number. So when we decompress pH to actual hydrogen ion concentration in soil, it exploded and it was about 72% variability. In other words, pH is compressed for a reason because acidity in soil does vary tremendously. I want to mention a wonderful study that's been ongoing in Missouri with the Citizen Science Initiative organized out of the Donald Danforth Plant Science Center. This was a recent poster that the participants in the study put together using our Solvita field test as a respiratory indicator among many, many different features that these students and adults working together across the state performed. Here was a classic result that they've had in the last 2 years, the rise of respiration during the summer and then the decline, tracking closely the temperature. So we've done this year after year and see this decline. The temperature is not directly controlling the respiration because the samples are being brought in and measured at the same room temperature. What appears to be the mechanism here is the warmer soil has more roots present and more exudates, so the microbe concentration is rising during the summer. Look at what they were able to discern from comparing cropping systems. This is prairies in response to rising temperature. Notice how contained and controlled the respiration is, and this is cropland. So I think out of a citizen science project, it's amazing that so much information that's meaningful is coming out of it, and you can log into their website and review these data. One thing that we are working on doing to help bolster confidence in methods and help train labs in moving into new biologically oriented tests is a soil health training and proficiency program that we've launched. In order to do this, we have made available to organizations and labs specially selected soils. Now, what we are doing is we're going out and getting soils with known management history and providing them to laboratories to use these different test methods. So we'll set you up with all the ingredients to do it, and then you report in a single blind sort of fashion. Now, our results last year with 32 labs across America, we sent them a depleted soil and an enriched soil. And the question was, could they distinguish them? And they did so with a high level of statistical accuracy. This was the depleted soil, and the range that all the soil labs got fitting a fairly normal distribution. And with the high respiration soil, you can see how the tendency is that, and this is common with just about any analyte, as the levels get higher, the labs start spreading out more, but it's still fairly well fit a normal distribution. And when we see a slight skew like this with slightly more labs getting a lower result than the average, we go in and we work on methodology. But we're very confident now in being able to say with this test at the given level of technique and method, we can accurately and statistically distinguish good from bad soils. And we're working now, we're sending out new soils this year that are a little bit closer together to see how close can we get and make this method work really well. So this is one of the really important and exciting things that has come out of the concern for proficiency and the concern for accuracy, that laboratories are now willing to work together and have themselves scored, and we can begin to build on every feature here that is important to carry this into the future. I want to just kind of in summary talk about what we have gone over today, and beginning with the whole area of soil health not being a thing in itself. It's almost a misnomer, and I have this statement on my own website saying soil health is a misnomer, and what I mean by that is it only exists and arises through the cooperation of plants and organisms, and that means it's under control of the farmer. Without the interaction of all these zones, this physical, chemical, and biological, you would have no soil health. So the most important thing that needs to be stressed here is that health is arising purely as an effect of a number of different parameters interacting together. Therefore, our path in testing becomes different than approaching it from the point of view of inorganic chemicals present in soil, which are put there and removed and then put back again. You can't think of soil health as something that's just put there. It's responding to the environment. One of the questions that somebody sent in prior to this webinar, a question concerned how can farmers encourage certain types of microbes in the soil? And what my response to that is the microbes will take care of themselves. You have to have practices that will bring them. And the most important thing actually is soil animals. We have farming systems that have greatly depleted soil animal population. And I'm thinking of not just earthworms, but insects and ants that are so important in the movement of detritus and residues from the soil surface into the soil. They are actually the primary decomposers. And we don't even have good methods to collect samples and measure them. Field biologists are good at this sort of thing, and the variability of these tests is likely to be high, but that would be no reason not to pursue it. One of the important things about soil animals is that they are the ones that are so significantly infected by our insecticide use. So bacteria and fungi, they're very tough and sturdy. Bacteria can consume and degrade just about any chemical we can throw at it, but the soil animals are more sensitive. So if we're going to build the populations of these soil health participants, we may have to start looking more carefully at the chemical usage and what it's doing to harm populations. So coming back to my strong theme here, there is no single test for soil health. Many people think my test, the Solvita test, is a single test for soil health, but it is not. It's actually a family. We call it a suite of tests. We developed one of them first and got it out of the gate, and we're grateful to all the labs that have worked with us on this, and also overseas now. But we always intended to keep moving to try to encompass some of the other aspects of soil. So respiration, the respiratory cycle in soil, is one of the most dominant features. And the beauty of the test is it assembles all organisms into one measurement. So a common feature of soil is insects, animals, microbes, bacteria, all breathing out CO2 and transforming carbon. Roots are also respiring CO2. So if you do studies in situ in the field, sometimes as much as half of the CO2 you absorb could observed could be coming from the roots themselves. So once you take the soil sample, bring it into the lab, that's no longer true. The respiration is the microbes consuming those roots and other litter in the soil. But this relates to the bigger picture of the buildup of CO2 in the atmosphere as well. And there's a tremendous amount of interest now in soil efflux studies that's not coming from a soil health point of view, but coming from a landscape ecology and climate point of view. And we're seeing a lot of papers now appearing from a different discipline that are very welcome, and I read them avidly because they're helping us shape these methods and develop them better. On the biochemical side, we've always felt we needed an amino acid or a protein or some kind of way of indicating this wonderful accumulation of organic matter that soils are capable of under good management practices. And if you can imagine that you have, as I showed that Flanagan series soil from Illinois, imagine having 1,100 pounds of organic nitrogen in the bank, so to speak, in your soil. That's a risk protection there in terms of good and bad years and changes in leaching of fertilizers put on. If we can find methods that help reveal this, it'll start shifting our perception of the quality of their soils. Finally, we have always wanted to do an aggregate stability test. Actually, I developed our aggregate stability test quite a long time ago and never found a mechanism or a means to release it. Last year, we made it available on a licensing basis to labs, and now 5 labs in the country are using it and 2 labs overseas. And we're beginning to see some really good indications coming out of making this test available. So ultimately, to build this picture of the triangle of results, we have to have multiple procedures. And I'm always looking for more methods. The laboratories are hard on us in the sense that they need methods that are cost effective. And that's why we make some of these innovations. I want to strongly emphasize again that different lab tests may reveal similar ranking. It's one of the most important things. I hear people always like focused on how accurate was the test, and I have to stand back and say, can you distinguish good and bad soil practices with a test? That's what we need to be able to do. And labs may get different results, and we keep working with them on this. But if the ranking of these farms and soils are similar or almost identical, I showed an example of the Woods End test being nearly identical in results to the Cornell test done differently, that means at least we have measurement systems that are effective in revealing these differences. We showed in the Illinois study extremely strong correlations between many tests across 6 soil families. And this is the hard work, folks, in terms of building soil health testing. We have to get to know our soils. It starts with soil taxonomy and the differences across a state, region, and climate. And we have to do more than just look at the topsoil. We have to become familiar with the entire profile. And our tests correlated with the work that Mulvaney has done on the nitrogen responsiveness of these soils. We're seeing that we can begin to build a model where the soil health test itself tells us something fairly strongly about the capability of these soils to respond or not respond to added fertilizer. Finally, in a Northeast study, and this is an ongoing study that will be hopefully expanded this next year, we have shown that soil biology tests actually varied less than nutrient tests on 17 dairy farms. Now, one of the thoughts coming out of this study is that possibly it has something to do with the fact that these farms all have manure in the system, and therefore we have a large static microbiome in the system that is relatively non-transient. Perhaps that helps make soil biology tests less variable, because it's hard to picture regions of a field that have a different microbiome, but it's easy to picture regions of a field that have different fertility levels because farmers spread manure unevenly, and this might have been a factor affecting the results, but they're very promising. And finally, I have worked strongly here to develop a proficiency process, so a means to help labs monitor themselves. One of the astonishing discoveries we made was how much soil handling affects the soil biology results. It's not an original discovery. I've now found evidence in papers going back into the early 70s, particularly by Bremner and Waring, warning us that if we do N-min and C-min tests, we have to be very, very concerned about soil handling, and it needs to be standardized. Today, there is no standardization of soil grinding and sieving in labs. And even though we all say we do it at 2 millimeter, I find one lab's 2 millimeter is really different from another lab's 2 millimeter soil. And there's been some defense recently, defending the industry practices of grinding and sieving. And we have gone back again and looked at some of the historical changes that have happened and find that in the early days we used to roll soils, and now everybody's grinding them. And it increases the throughput. But is grinding the right approach when you're doing soil biology, when you're measuring the activity of microbes in soil? We have to be concerned that we've changed the environment to the extent that we're not able to properly observe it. So on the good news side, and some studies I've cooperated on with, and we did an NAPT comparison study last year, we can get reasonably accurate results with soils that have been ground by one grinding method if we're very, very careful with how we moisten the sample. So we have learned a lot in this study, and labs need to be prepared to focus more attention on it. In releasing the VAST test here into laboratories, we had to point out to them that you can no longer grind a soil to do an aggregate stability test. We do sieve it to 2 millimeters, but one of the reasons we've seen early variability in these tests is our difficulty in controlling how laboratories are handling samples. So we all have a lot of work to do. Farmers have a lot of advancements to make in practices, and the scientists in the labs, I say you have the same challenge too. You have to change some of your practices. If we are going to help launch soil biology testing and have it grow and mature and become a robust area of evaluation, we may have to make some deep-seated changes in our laboratory processes. Our company, Woods End Laboratory, and staff are available to work with anybody who wants to receive training and to get into the field. We have relatively simple methods that you can add to complement your chemistry work, and we will train you in doing it and then invite you into the proficiency program so you can monitor your results. So it's a work in process, and it continues to interest me and everybody I work with, and it seems like there's no stopping it. I want to prepare to stop here and to give some time for questions and answers that have come up. So I'm going to mention a few that were sent in to me prior to this presentation. Somebody asked, how do we measure mineralizable nitrogen and sulfur from organic matter? And I think I've touched on this indirectly throughout my presentation. In order to do this accurately, you have to know the rate of decomposition, and that's why we do respiration studies. We're really looking at the change of carbon. But then, more accurately, you have to know the content of nitrogen and sulfur in the organic matter, and then the mineralization rate times the mass of nitrogen and sulfur will give you your potential mineralization. And then the curveball in all this is what I call re-immobilization. As fast as some of these nutrients are mineralized, the soil may re-immobilize them. And it's getting more challenging. The more cover cropping we do, the more litter there's in soil, the more immobilization is happening, the more you have a bionutrient and not a soil nutrient. So that's one question that was very interesting. Another very interesting question, are soil health tests being correlated with high organic matter soils? Now, you did see some evidence today from some of those Illinois soils that were 6 and 7% organic matter, that we have been looking at relatively high organic matter soils, but they are problematic in a number of ways. They release huge amounts of CO2 that we're not accustomed to. peaty soils. In the United Kingdom and New Zealand, we're working with soils that are pasture soils, high organic matter, huge amounts of respiration involved. So yes, we have to alter the method. So the issue of long-term yield trends came up in some questions sent to me. I think I've shown really good evidence that by accessing long-term studies, long-term crop rotation or field plot studies, we can build this relatively quickly. I'm going to throw it open now to the moderator if there are any questions that that have come in or we should deal with in the remaining few minutes of our time.
Thanks so much, Will. Yep, we have a few other questions, and I will just answer or ask one. Have you utilized and/or compared plant tissue analysis to go along with soil analysis? Are there individuals who can use their senses that can analyze soils in this manner that compare well with laboratory analysis?
When you say plant tissue, you mean just crop tissue analysis? I think that's
I believe so.
Yeah. We have certainly been doing that. Every year I do field plot studies here on our research farm. So, you know, if you look at this dramatic picture on the screen here of a cross-section of a field of potatoes, a tremendous amount of the nutrients are split between the tuber and the green foliage on top. And we've been tracking the accumulation of nitrogen during the season by taking periodic sample points. So yes, I'm a big believer in tissue tests belonging in the picture here to help us understand the accumulation of nutrients and relate it more closely to what's going on in the soil. So if you could do an ideal study, you'd measure it every week through the season, take your soil health indicator, make sure you get your temperature of the soil because that's the big driver. The moisture is not as strong of a driver under field conditions, and then do your tissue testing.
And I've got one more. How does bacteria change soil bulk density?
Well, bacteria, fungi, soil animals change it indirectly. First of all, by nature of being an organism comprised largely of organic matter, the density of bacteria and fungi is very low compared to soil. It's an interesting question though, because what is the mass of these organisms in an acre? And I've seen anywhere from a ton to 10 tons. So if you have 10 tons of organisms, that will definitely reduce the bulk density. But don't forget, an acre of soil, 6 inches weighs 1,000 tons. So the effect is indirect. The effect is that the bacteria and fungi are there because organic matter is present that's being built into aggregates. And it's the aggregates that are increasing the volume of the soil quite substantially. So a soil can have a different volume density by a factor of 2, going from 1% organic matter up to 5 or 6% organic matter.
Okay, and last question. Has the repeatability of Solvita tests been evaluated in soils with different textures?
I think I addressed that really well today. We've been looking at soils all over the country on these studies, and texture, it's not really texture that's the matter, it's the structure of the soil that's so important. If we grind soil and destroy the structure of the soil, we'll still have the same texture, sand, silt, and clay, and the relationship of them, but we've destroyed the microaggregate structure, which is the supportive framework of respiratory studies. So the evidence is out there that once you destructure soil, you've changed the nitrogen mineralization potential quite a bit because you've exposed protected organic matter, and the exposure of protected organic matter within the structure of soil needs to be addressed more closely. So it's an ongoing concern. Repeatability is always of interest to us, and as I have noted in the program, you can participate with us in a proficiency program in which we've specially prepared the soil based on its structural characteristics. Thank you very much.
Great, thank you, Will. That concludes our webinar on soil health challenges and measurement for today. If anyone has any more questions, please reach out to Will via his email at will.brinton@woodsend.com. You will find this webinar and other soybean and agronomic resources on the checkoff-funded website, illsoyadvisor.com. Thank you for attending and have a great day.